Hello everyone, this is BrainMap and I'm Chihan, co-host of the BrainMap. And this seminar series is co-sponsored by P41 funded Center for Mesoscale Mapping housed in the Martinos Center. Today we'll have great pleasure to have our speaker Ali Gholipour. Ali Gholipour is Associate Professor of Radiology at Harvard Medical School, Director of Translational Radiology Research, Director of Intelligent Medical Imaging Research Group, and a member of the Computational Radiology Laboratory in the Radiology Department at the Boston Children's Hospital. He received all his degrees in electric engineering and is a senior member of the Institute of Electrics and Electronics Engineers, and a member of the editorial boards of Medical Image Analysis and IEEE transactions on medical imaging. His research interest lies in the broad area of machine learning in medical imaging with a focus on magnetic resonance imaging for the analysis of early human brain development. Thank you, Ali, for having a great talk today, and the stage is yours.
Hello, everybody. Good afternoon. I would like to thank the organizer for inviting me and to all of you for participating in this talk. Happy New Year. I wish you all the best in 2023. I will talk about some of our technical work that is towards mapping the fetal brain development in utero. That means imaging live fetuses. This is the outline of my presentation. I will start with an introduction to the significance and background in imaging early human brain development, specifically with a focus on prenatal brain development. Then I will talk about the techniques that have been developed over the past decade, specifically motion robust super resolution fetal MRI, motion robust diffusion weighted MRI, fetal brain MRI atlases, and some post-acquisition processing techniques that we've developed. And then I will also mention some applications of it. From the 4th week of pregnancy until mid-gestation, which will be around 18 weeks, an embryo's brain generates neurons at an astonishing rate of almost 500,000 per minute. Approximately 100 billion neurons are generated, and 100 trillion is the minimum number of neural connections that are estimated to exist in the human brain, or the synapses that form. Ultimately, the mesencephalon and the telencephalon takes shape as a very complex living organ. Fetal imaging has been instrumental in shaping the field of fetal neurology or neuroscience. We have 3D ultrasound nowadays. You see examples of ultrasound images. Fetal MRI has also been widely used in the past decades. A little bit about the history of fetal imaging. This is the first ultrasound in the fetus that was reported in 1968. And from there, you see that over a period of about 15 years or so, or 20 years, we saw 3D images of the fetus. This was first reported by Dr. Baba, who was an MD and a PhD in electrical engineering. And then we see in 2000s, nice 3D and 4D ultrasound images, and fetal MRI has also advanced. Compared to ultrasound, MRI offers a lot. It provides really advanced and complex contrast mechanisms that let us to look at not only the brain structure, but also the structure of vasculature, the microstructure of the brain connections, and also the brain function. However, most advanced MRI techniques are long in terms of image acquisition, and therefore they are very susceptible to subject motion. And fetuses move a lot during MRI acquisitions. So you can imagine that that would create significant challenges for our techniques. Because we are trying to scan a moving subject for which the controlling motion is very difficult, if not impossible, we try to get the fastest MRI acquisitions that are available. And among them, a sequence that gives us relatively good contrast is a half Fourier single shot fast spin echo scan. Even though these acquisitions are pretty fast in terms of the readout time, but occasionally the fetuses move so fast that the signal is completely disrupted and the encoding for MRI image reconstruction fails. These are examples of failed acquisitions. However, many times fetuses don't move too fast. If they move to a good extent while they're in the amniotic fluid and don't have too much space to move quickly and in large angles, that technique, which I will refer to it as single shot fast spin echo or SSFSE, would work to acquire 2D image acquisitions. If we acquire a bunch of those images, basically covering the anatomy in 3D, and these are fast 2D slice acquisitions, every slice looks good, and these have been cropped around the fetal brain. You see the quality of the images are nice, but when you stack them and look at them from the out-of-plane views, in other words, this was an axial acquisition, you look at them from the coronal and sagittal views, you see that because of motion and thick slice acquisitions, these are quite disrupted. There's no coherent anatomic boundary in these images. So this is how the images would look like. That brings me to discuss the first technique that we developed about more than 10 years ago on motion robust super resolution imaging. So basically we asked this question, given a number of those stack of slice acquisitions which are disrupted by thick slices and motion in between slices, can we reconstruct a 3D image that looks good and shows the anatomy with coherent anatomical boundaries in all three planes? And the answer was yes. So we designed an algorithm that took those slices, basically a number of 2D slice acquisitions of MRI could be in any angle, any direction, and then by doing interslice motion correction or retrospectively, we could bring those slices into realignment and solve an inverse problem to reconstruct the volume. And hopefully that volume would look like this one. If we just take a bunch of those images and average them, we'll end up with an image that is blurry like this because of uncompensated motion. However, we showed that if we go through iterations of our algorithm and generate an image, use that super resolution image as a reference volume, and again, use that to guide interslice motion correction by slice to volume realignment or registration, then through a few iterations of that process, we'll get a really nice image that looks good in the three planes. How did we do that? We said that if we first wanted to have a forward model of how slices are acquired. We assume that there is an imaged volume, basically the fetal brain in this example, that undergoes motion in 3D space. Slice selection is done through the slice excitation, and the slice profile is taken into account. Then MRI signal is acquired, quanta is sampled, and that gives us an image. If we can accurately model this acquisition, then we can solve the inverse problem, and we opt to use robust estimation using M estimation to solve that problem. So basically, this will be our forward model. Slices acquired, and X is our hypothetical super resolved image of the anatomy. Why do we use robust estimation? Because occasionally, as I mentioned, some slices are affected by motion because of fast motion, and the technique should be robust and be able to filter those artifacts and those artifactual slices. Also, registration may not always be successful. Sometimes it fails. So we want the system to be robust. This is an example outcome of the algorithm for 26-week fetus. In the first row, you see an image acquired in the sagittal view with respect to the fetal brain. In the second row, you see that we have averaged some of those slices, axial, coronal, sagittal acquisitions. The image is pretty blurry, but after those iterations, we get a really nice image looking good in all of the planes, in all of the slices. So this is basically a coherent 3D anatomical image of the subject that shows us the details of the anatomy that we were not able to see without having this technique. More importantly, that led us to do some of the first analysis of the cortical analysis, let's say cortical development analysis, circle depth quantification in the fetus. Remember that with those images that were not good in the out of plane views, that didn't provide coherent anatomical boundaries in 3D, this type of analysis was not possible. So it first became possible when we developed that technique. This line of work on anatomical reconstruction of fetal MRI from slice to volume registration has advanced since then. A couple of years after our work, there was this nice other paper by a group in London, my colleague. They enhanced the algorithms by intensity matching and a more accurate implementation of the slice profile. There have been more recent works, including this work by another colleague of mine, Bernard Kynes, who developed a patch to volume reconstruction approach that worked well even in the fetal body area. Of course, even newer versions of those techniques based on CUDA implementation on GPUs let us to do fast reconstructions, and we see even newer papers in 2020, and these algorithms are developing. This is another work by another colleague of mine, Michael Ebner and colleagues, who developed a framework based on Python, and this directly reconstructs an image based on slices in our atlas space, and I will talk about the atlas space as well. So in the meantime, while the field is progressing in that front, we tried to work on diffusion weighted MRI because, as I said, the main advantage of MRI over other prenatal screening tools is the subtle contrast that it provides. Ultrasound cannot show us in these anatomical details or the microstructure, connectivity and function of the brain, but MRI can. However, as you can imagine, those even more advanced techniques of MRI, functional MRI and diffusion, are even more susceptible to motion. How do we compare diffusion MRI with the T2 structural MRI? It's based on echoplanar imaging, so it's faster. There is a base image, non-diffusion sensitized, and there are some diffusion sensitized scans. The advantage of this sequence compared to T2 is that it has a higher sampling rate, 10 Hertz, because of echoplanar imaging. 10 slices per second, it's roughly 10 to 15 times faster than our T2 weighted slice acquisitions. So hypothetically, slices should be less affected by motion, but there are also some disadvantages, such as the varying contrast due to diffusion sensitization, lower resolution, lower signal-to-noise ratio. If you just get a diffusion weighted image of a fetus of the scanner, that is the fractional anisotropy map that you get. Pretty noisy because of the uncompensated motion. What we did, we tried to take advantage of that higher sampling rate of slice acquisitions to enhance our slice to volume registration while we were sort of challenged by the fact that these diffusion scans had lower resolution and lower SNR. So somehow we tried to use the faster sampling to compensate for those challenges. For that reason, we developed this was an innovative idea of doing registration-based motion tracking. So basically embedding registration into a Kalman filter in which we modeled the state of the position of the fetus as the state variables with 6 parameters, and then tried to solve that state estimation problem for sequential motion estimation. And this is based on real motion experiment in an adult volunteer where we could really measure motion with a sensor. This is the actual ground truth motion. And this is if we were just doing sequential registration. However, with our robust Kalman filter-based motion tracking, which I would call it MTSVR for motion tracking based slice to volume registration, we were able to track motion of that volunteer pretty well. So it's really capturing the dynamics of the motion of the subject by Kalman filtering and image registration. Now, for diffusion imaging, once we calculate those motion parameters per slice, and really the resolution of a motion estimation through MTSVR is at the slice level, then we will end up having variable length diffusion measurements at every voxel of the image. It's no longer like we have a fixed size number of measurements with volumes that are representative. Now it's a complex data structure that has variable length diffusion measurements with rotating gradient vectors that are per voxel. And so we really need to solve it at voxel level and also take into account that that is a really scattered data interpolation, taking into account the slice profile of the EPI acquisition for diffusion imaging. So we formulated that, and we had these alpha terms here. Weighted linear least squares is the approach we took in order to fit a diffusion tensor model at every voxel to that model. And of course, we experimented with that and tried compared to different computation techniques for diffusion tensor imaging. Now, this is again an adult volunteer in which we know the ground truth. When the volunteer didn't move, you would get this type of fractional, color fractional anisotropy in the midbrain, the area of the corpus callosum, and some corticospinal tracts and other tracts passing. Now, if you have a motion corrupted image in which the volunteer moved, and you apply the conventional algorithm, you will get a very disrupted image because of uncompensated motion. If you do volume to volume registration, for every volume of those diffusion MRI scans for that same motion data, you will get an image like this. And with our algorithm, you'll see that you get much better mapping compared to the ground truth. We published these works in around 2016 and 2017 and applied it directly to fetuses. On the top left, you see an image of fractional color fractional anisotropy for a fetus with a conventional approach, linear least squares, and we have different variations, including weighted linear least squares with the conventional approach. Again, didn't work. Then with volume to volume registration and weighted linear least squares, a little bit better. Now you see the corpus callosum in the sagittal view. But if you apply our technique, motion tracking based SVR with weighted linear least squares, now you get the best image. Cingulum, corpus callosum, and many other structures are now visible on that. I said that fast motion can affect the slices. So in our T2 weighted imaging, we tried to be robust to those by using M estimation. In diffusion imaging, we tried to find those corrupted slices by machine learning. And the reason behind it is that no longer statistical approach was the best approach to find those slices. It is quite possible that because these are quite fast acquisitions, every one of these volumes is recorded in 3 seconds. A statistical approach that doesn't see the majority of slices either corrupted or not corrupted would fail. But we showed that by using a support vector machine, training it on a bunch of annotated motion corrupted slices, we were able to detect those slices and really remove their effect from the final reconstruction for DTI estimation. We did some quantitative analysis. The main idea behind this analysis was that in fiber-rich regions with single fiber pathways, for example, in corpus callosum of the limbs of the internal capsule, we expected really high FA. And our assumption was that if there was uncompensated motion, as you see, they result in blurring, they could result in reduced FA because, for example, in corpus callosum, the regions around it, ventricles and other regions, have lower FA, so that would reduce the FA in the corpus callosum if the motion is not compensated well. So basically, we assume that if our motion compensation is effective, we will see higher FA values in our images. And it was confirmed These circles, black circles, are the conventional approach. The squares are the volume-to-volume registration. We have the slice-to-volume registration and motion tracking with slice-to-volume registration are ultimate solution for fetal diffusion-weighted imaging. And you see that we got consistently highest FAs for the corpus callosum LIC using our approach. Also, when we looked at a bunch of fetal DTI images reconstruct through that approach, we saw that as age increased for those fetuses, we saw an increasing value for FA, which is confirmed by our knowledge of how the brain develops in the corpus callosum region and also in the LIC. We expect the FA and anisotropy as fibers form in those regions to increase constantly during gestation. We looked at the values of the FA as a function of the average absolute motion that we measured based on our slice realignment metrics in the fetuses, in roughly a number of fetuses, 20 or so, and also FA as a function of the, in the LIC as a function of the average absolute motion. And in both cases, we saw that as the motion, absolute average motion increased, the conventional and VVR methods performance dropped, but our techniques performed robustly with respect to the motion. Then we looked at average speed of motion, how fast the fetuses moved for each TR, and also looked at the FA in those two regions. This time, every technique, including ours, showed a performance drop, but of course, ours was still much more robust than the conventional approaches, still effective. Based on those results, we expected good trajectography in the fetus, but we actually ourselves were surprised to see these many connections or fiber pathways were detected in fetuses as young as 26 weeks. You see a lot of structure in those. So those strategies or technologies that we developed over those years led us to do a lot of work. For example, we realized that resources that existed for neuroimaging for adults and adolescents and children, newborns, didn't exist for fetal subjects. So we tried to address those unmet needs. And based on the super resolution T2 weighted image reconstructions that we had from 18 normal fetuses scanned between 21 to 39 weeks, we were able to build a spatiotemporal fetal brain MRI atlas. Now this atlas is a normative atlas of MRI, shows how the brain is developed across gestation. Of course, these are only representative slices of the atlas, but the atlas can be generated at every fraction of gestation age from the input images because it's a continuous spatiotemporal atlas construction framework. We have released the atlas at every gestation age at 1 week intervals on our website for download by the community. The atlas over the years, through the help of my great colleagues and significant contributors who have been listed here, we were able to refine and draw masks of different tissue types, transient brain compartments, deep cortical gray matter structures, and even parcellations on the fetal atlas. And those are all available along with the atlas that would be helpful for studies of the brain connectome in the fetus. Similarly, along with our structural brain atlas, we tried to use our diffusion MRI reconstruction technique to build diffusion atlases of the fetal brain. Could we do that? This was more challenging. Our initial attempts failed, but then we realized that if we had a framework for tensor to tensor registration, really robust framework with the unbiased group averaging, we were able to map the images, basically weighting that averaging by the gestational age within a window of 1 week, symmetric around at the time that we want to reconstruct an atlas. And this was a great work done by Dr. Khan at that time, published in Neuroimage. And these are snapshots of the diffusion atlas that we generated. This is also available on our website along with the other atlas for diffusion for the T2 weighted images of the fetus. Now, based on the atlas, we were able to find a lot of structures that you can see. that depict the microstructure of the brain. You see that from early on during gestation, 27 weeks, the brain is very smooth and still a lot of structures are seen. How much it changes to ultimately closer to birth at 38 weeks, basically now has a structure that looks alike, very much like the adult or mature brain, at least in terms of the structure. Of course, a lot of myelination and other development has to occur after birth. Now, another colleague of mine, Dr. James and colleagues, in particular, Camilo Calixto, had made segmentations on our diffusion atlas, and this is an ongoing work. The paper was just published in Human Brain Mapping. Now, this is a snapshot of our atlases, parcellations, diffusion, and these parcellations will enable working on the seed-based or region-based connectome analysis for the fetus. Now, the last part of the techniques that I would like to review are post-acquisition processing. So basically, based on those developments, one can come up with really advanced fetal MRI processing pipelines, such as this one. In the first box up here, we have a structural T2-weighted image processing, basically starting with the slices that are disrupted by motion. You have an initial volume, which might be blurry, but through super resolution reconstruction, you get a really nice image. Then we brain mask it. We have automatic tools for that now. I will briefly talk about those. And for bias correction, then register it to the atlas coordinates. And that is not an easy task either, because these fetuses can be in any orientation. Once you have that, you can use multi-atlas segmentation or deep learning or whatever technique that you have in order to find the parcels and segments on that brain. In the bottom box, you have the diffusion processing. Basically, you have the B0s and diffusion sensitized images, motion tracking slice to volume registration. And again, once you have the color phase, with the use of that and the parcellations, you could look at whole brain tractography and ROI-based connectivity analysis. As you see, there are a bunch of processing that is needed through that process, including several segmentation tasks. So once we have that pipeline, we were asking ourselves, can parts of these processing be automatized to improve both the acquisition and processing part? And we were not only trying to improve the processing, but we also had a look at acquisition in order to inform the acquisition to make it even better. So early on in 2017, we came up with deep learning-based fetal brain extraction on 2D slices. So basically, you see how that technique works. It's an auto-context unit that we developed at that time. And basically, well, this video doesn't work, but I have great examples here to show you. Basically, very challenging cases that we've shown here. And this row is, or ground truth, is manual segmentation of the brain parenchyma by an expert. In the first column, we have our unit at that time. And there are a couple of other techniques that, deep learning technique that we implemented, and another post-processed version of that with Markov random fields. And you'll see that for an abnormal brain, very abnormal brain shape, which was not even an example or a similar example to this was not in our training set for the deep learning model. The model was surprisingly, even to our surprise, able to find the brain parenchyma, the part of it that existed and was not affected by this abnormality. And for cases that there was a lot of motion, our expert decided not to find this part of the brain, but our algorithm even tried to find that part of the brain too. And as you see from the out of plane view, it did a pretty good job in not only finding the brain in good slices, but those slices which were affected by motion, it did a decent job in finding those. In terms of DICE values and other performance metrics, we performed very well with the time that was considered real time, given the 1.5 second acquisition time for T2s compared to the other algorithm, including very sophisticated algorithms based on SIFT, random forest, and conditional random fields. we also recently looked at different configurations of deep learning networks, and we have deep learning models now that can segment the brain, basically detect and extract the brain in fetal MRIs in about 3 milliseconds. And this is only possible if you use deep learning approaches. I'm not aware of any other image processing technique other than not using deep learning directly, and to be able to do that with that speed and with such a high accuracy. Another part of the processing was to register these brains into the atlas. And sometimes that can be a very challenging task. As you see, these are brains of different ages, with very different shapes. They can be in any arbitrary orientation, unlike adults or other subjects that you put in the scanner in a certain direction. You know that at least the angles of the brain should be in certain positions. Fetuses can be in any orientation with respect to the scanner or word coordinate system. So that can be a challenging task. Again, by using deep learning and merging it with registration, we showed that we are able to register our images to the atlas. Basically, this is volume-to-volume registration initialized by deep learning for pose estimation. And this is a snapshot of our pose estimation, 3D pose estimation. It's a regression neural network that learns from a bunch of examples that we create based on real images to find the orientation of the fetuses. And we extended that work to even do deep predictive fetal motion tracking in the fetus. Now, other parts of the algorithm for the purposes of analyzing cortical folding, doing connectivity analysis, region-based volumetric and morphologic analysis, we wanted accurate segmentation techniques. And in more recent years, we took advantage of the deep learning frameworks based on attention and other mechanisms that were showed promise in the computer vision community, and looks like it was pretty good. So with our technique, we have cortical plate segmentations generated automatically that match to the ground truth manual segmentations much better than alternative techniques. And this was published in IEEE TMI in 2021. I will skip the details of those, but just in terms of the performance metrics compared to other networks, alternative approaches, we got much better dice and 95% Hausdorff distance and other metrics. And of course, we did expert evaluation of multi-attach segmentation based on those, and those showed that our technique performed much better than the others. We try, in our most recent works, we have developed deep learning techniques to segment not only cortical plate, but also other tissues of the fetal brain, and the paper was published in Medical Image Analysis this year. Now we try to process diffusion weighted images also the same way. This was one work that we did in order to do group analysis in fetal cohorts using the reconstructed diffusion tensor images, and we used tract specific type analysis, basically apply a robust DTI estimation, and then study specific unbiased regression to the template reconstruction. And through that, we were able to robustly do tract segmentation and medial tract computation for tract specific analysis, which showed us differences between fetal cohorts. We showed in recent years that deep learning also can improve the diffusion tensor estimation for fetal MRI. So basically, if you're given, these are newborn subjects from the Developing Human Connectome project, in which we have motion-free images with relatively dense Q-space measurements. In order to simulate the fetal scenario, we tried to do a much lower number of measurements with lower SNR, simulated that, and applied weighted linear least squares, non-linear least squares, and restore approaches to fit the diffusion tensor model to those images. They failed because of the low SNR, but we showed that our proposed deep learning algorithm performed well even in the presence of those things. So then we tried to extend that to the fetal case, and so to give you a sense of how it would look like, newborn images, DHCP quality, you have 88 measurements with 3 shells at least, and SNR is about, estimated SNR is about 15 to 20 dB, whereas in our fetal, first of all, depending on the motion and the position of the fetus, it can change drastically between fetuses, but the number of measurements can be between 12, number of useful measurements can be between 12 to 48 or even more if we spend more time, but still SNR is very low. We tried to model that, learn from the gradient tables and learn from the noise distribution in the fetal to train a deep learning model to estimate the diffusion tensor model per voxel for the fetus. And the results showed, first of all, were verified in the newborn scenario. And in the fetal case, on the left, you see the original estimations with our non-deep learning based methods. And now with the deep learning based methods, we get much better acquisitions, even in the presence of significant motion artifacts in those fetuses. And these were published in a few papers that were looked at different types of diffusion weighted image modeling, including fiber orientation distribution functions based on DWI. Now with that, I can talk about the applications, and I will try to be quick on this. I have to say that most part of these works were done by our great colleagues, and we were fortunate to have collaborators throughout this project, which helped us to assess the utility of these techniques. First of all, out of our atlases, we were able to segment the fetal brains and look at really how normal values for the cortical plate volumes, cerebellar volumes, brainstem volumes were changing. You see that they're still in the exponential phase of growth during these late gestation weeks. So these were new data that were possible to achieve because of the advances in technology that we had. That also led our colleague to look at altered brain development in congenital heart disease, really looking at the sulcal depth, and you see where they found differences between fetuses with congenital heart disease and normals. Another most recent paper based on comparing volumes of brain structures in congenital heart disease patients and controls was done by my colleagues, Dr. Rollins and Athinoula, publishing Annals of Neurology, finding differences in total brain volumes and in the volume of the ventricular zone between, now you see the control fetuses in red and fetuses with congenital heart disease in blue that have obviously showing lower average ventricular zone volumes. Now, my colleagues published and did some studies and published other works reporting the great results of those studies that gave us insight into the pattern and trajectories of brain development in congenital heart disease, including the work that my colleague Dr. James did in looking at the fetal brain maturation scores in those patient cohorts. Another colleague of mine, Dr. Vazunk, has looked at cortical plate and subplate in 22 regions, 42 healthy fetuses during gestation, and basically quantitatively looked at regional sex differences between fetuses. Differences were reported in volumes of the inferior frontal gyrus in the frontal lobe, limbic lobe, and occipital lobe. There were also hemispheric asymmetry that was discovered by running these techniques, and of course, spatiotemporal differences in the regional cortical plate and subplate volume growth compared to the other regions that also gave us insight into how the brain develops during those ages. And of course, she also did some work on cortical development and gene expression, basically looking at the association between quantitative MRI markers of cortical reorganization based on the transient fetal compartments and the gene expression using human prenatal brain development data, which identified the genes that were showing higher association and correlation with those brain development markers. My colleagues were able to do tractography, seed-based tractography in different regions of the brains in fetuses and also quantify the volumes of different regions, for example, the middle cerebellar peduncles, which is in charge for the cognitive and motor compartments of the brain, looking at how the fractional anisotropy and ADC change during gestation for normal fetuses during those ages. And of course, we were able to see the pattern of development of major fiber bundles using diffusion tensor imaging based on our atlases and the analysis that my colleagues did. With that, I would like to conclude. In summary, motion robust super resolution fetal MRI corrects interslice motion and improves image resolution, provides us with images with coherent anatomic boundaries in 3D. This enabled motion robust analysis of diffusion weighted MRI and also functional MRI, which we're looking at. It enabled atlas construction, automatic segmentation of the fetal brain, which enables many quantitative analysis, enabled longitudinal and group analysis of normal and abnormal growth. And I showed you some examples of the work that my colleagues did on that. Deep learning enabled improved processing of fetal MRI scans, including brain extraction, tissue segmentation, and registration. We also took advantage of deep learning to improve diffusion parameter estimation. I'm fortunate and was privileged to work with a lot of colleagues, great colleagues and collaborators across institutes. And of course, this work was not possible without their help. There are actually more than this number that I could put on this slide, but I express my sincere thank to all of my colleagues for the work that was done. And of course, sources of funding and support that we received from different institutions, including in particular NIH. If there are any questions, I'm happy to take.